Hello everyone. Today we are going to start with the external morphology of Herdmania. So now let's discuss the shape, size and coloration. Body is laterally compressed, somewhat oblong or rectangular in shape, attached and slightly narrower than free end. Okay, so let's see here. Herdmania is something like this in shape with branchial and atrial, branchial. Here it is branchial and atrial siphon. Okay, so body is laterally compressed. Laterally compressed means compressed, it is compressed from the side. Suppose these are the sides, so it is compressed like this. If we push these two sides towards the inner side, then it will become laterally compressed. So it is laterally compressed body, somewhat oblong or rectangular in shape. Okay, let's see. If I say this is rectangle, okay, so body is somewhat rectangular in shape. It is laterally compressed. Laterally compressed किया इसको. Lateral means side से अगर compress करेंगे तो ये body कुछ ऐसी हो जाएगी. Edges will be destroyed. Okay, so this is the shape of Herdmania, that is laterally compressed. Somewhat it is triangular and oblong in shape. Okay, rectangular, sorry, not triangular. Attached end is slightly narrower than free end. Attached end is the foot region, which is slightly narrower than the body region. This is the body region, this is the foot region. Foot region is entirely composed of test, so this is slightly narrower than the um, free end. Okay, attached end, that means foot is slightly narrower than the free end. Now next point says average adult measures 9.5 by 7 by 4 centimeter. This is the size of the average adult. Foot when present is 3 to 4 centimeter long. Foot when present, क्योंकि in the last lecture I have already discussed that foot is sometimes it's absent and sometimes it's present depending upon the substratum to which Herdmania is attached. So size increases with age. Okay, obviously it will increase with age. Okay, size increases with age. Old individuals up to 13 by 8 by 4.5 centimeter. This was all about the size of average adult and old individuals. Now fresh specimens pinkish in color due to the distribution of fresh specimens pinkish in color due to the distribution of blood vessels in the superficial area of test. Okay, so fresh specimens, that are the fresh specimens which are collected from water bodies, they are pinkish in color due to the distribution of blood vessels in the superficial area of test. Test is the outermost covering of Herdmania. So pinkish color kahan se aa raha hai? Due to the distribution of blood vessels in the superficial area. That is superficial मतलब जो ऊपर का above area जो होगा इसका, यहाँ पे this is pinkish or pinkish due to the distribution of blood vessels. So it shows scattered bright red patches due to the vascular ampullae, a characteristic feature of Herdmania. Now this is very important. This is the characteristic feature of Herdmania. Okay, let's see here. Okay, if you see here, suppose this is the structure of Herdmania. Let's make it more clear. Okay, somewhat this is the structure of Herdmania. Okay, this is the foot region, this is the foot region, and this is body proper. This is body proper. Okay, this one is test. It is entirely covered by test. Okay, so it says that shows scattered bright red patches like this. patches are present of red color. Okay, red patches are present on the surface of Herdmania, that is in the test. It is present superficially, which is the characteristic feature of Herdmania. So if you see here, these are the red patches. Red patches कहाँ से आ रहे हैं? Okay, red patches कहाँ से आ रहे हैं? Red patches are coming from the blood vessels due to the vascular ampullae. So let's see here. Suppose uh, these are the blood vessels. Okay, so the terminal region, the end region of blood vessels gets swollen. So this region is now known as ampullae. Okay, this region is known as ampullae. This swollen region is known as vascular ampullae. So blood vessels are present in the test. These blood vessels get swollen at their terminal ends. This is the terminal end to form vascular ampullae. Ampullae is the swollen region. So that is the reason that red patches are visible on the surface of Herdmania. So let's proceed further. Now next slide is the division of body. I previously told you that body of Herdmania is divided in two parts. Okay, this is the body of Herdmania. Suppose this is the foot region, below region is the foot region, and this is body proper. Okay, so this is body proper and this is foot region. Now body proper is the distal free part, which is this is the foot region, as I previously told you, and this is body proper. Now this is attached to substratum. Okay, now its distal part is this one. This one is distal region and this one is proximal region. Okay, this one is attached to the substratum. It is attached to the substratum here, which is the foot region, and this one is free region. So free region is longer than basal part or foot and broader. Okay, free end is drawn into two short cylindrical projections called branchial and atrial siphons होते हैं. ठीक है, दो projections होते हैं body proper में, that is the distal free end, this one and this one. This one is branchial siphon and this one is atrial siphon, which is present on sorry, this is the branchial siphon and this is atrial siphon. Branchial siphon is present on anterior end and atrial siphon is present on the dorsal side. This one is branchial siphon, which is smaller, up to 1 centimeter long, 1 centimeter long and directed outwards. It is directed outwards. Sorry, it is directed outwards here, and it bears a terminal opening which is known as branchial aperture or mouth. Okay, so this is the incurrent opening. Incurrent opening means we have studied further that water current enters through branchial siphon and it exits through atrial siphon. Okay, so here it is the atrial siphon, which is directed upwards. It is directed upwards and it is 1.5 centimeter long, which is stated here. Okay, this one is known as excurrent opening, that I have already told you. Each aperture is guarded by four distinct lobes or lips formed by much elastic test. Okay, each aperture, this one is the apertures. These are both, each aperture is guarded by four distinct lobes or lips. It is formed like this. Okay, these are the four distinct lobes or lips which guard both these siphons. Okay, so these are made up of test, entirely made up of test, and these structures they both are guarding the branchial and atrial apertures. Okay, let's proceed further. Now is the foot. Foot is entirely made up of test, dirty in color and rough due to sand particles, shell pieces, and other foreign bodies attached to it. This is the foot. Suppose this is the foot of Herdmania and this is the body. So it has dirty in color, it has rough, it has sand particles, shell pieces, and other foreign bodies attached to it. Okay, I have told you in previous lecture that foot might be present or it might be absent. So, when herd mania gets attached to some soft substratum, okay, so if the substratum is soft, such as sandy bottom, so the animal anchors its body by extending a foot. Foot is narrow and shorter than body proper. Okay. Now, if herd mania gets attached to a hard substratum such as rocks and we can assume wood like something. Okay, so this body proper becomes attached by forming a broad, flat or concave base, and then foot is absent. Okay, if this is the, suppose this is the rocky substratum, so Herdmania gets attached by its body by forming a broad concave base. Okay, flat and concave base, and foot is absent. Foot gets, what we can say, contracted. Okay, so foot is absent completely and the body gets attached directly to the rocky substance or rocky substratum or mollusk shell, can we can say that. Okay, and here it is that foot acts as a balancer to keep the body erect when detached. Now when this herd mania gets detached, sorry, when it detached gets detached, then in order to balance the body, this foot helps. Foot acts as a balancer, like in our case, as our ears act as balancer of the body. Okay, and stetocyst is present in the organisms for body balancing. In the same way, foot keeps the body erect when detached. Okay, it acts as a balancer in order to keep the body erect when it is detached from the substratum. Okay, so next one is test or tunic. Okay, body of Herdmania, it is covered by test, entire body including the foot. Foot is also covered by test. Now this is the anterior, sorry, this is the body proper and this is foot. So test covers the entire body of Herdmania including foot. Now it is protective in nature. Okay, it acts as a accessory respiratory and receptor organ. Receptor organ कैसे बनेगा? It is it has vascular ampullae, we have studied just now. Okay, so these vascular ampullae also have nerve endings. So as a result, it acts as a receptor organ and respiratory organ क्यों कहेंगे इसको? टेस्ट को एक्सेसरी रेस्पिरेटरी ऑर्गन होता है क्योंकि vascular ampullae are present here. Ampullae is the swollen region. It contains maximum supply of blood. Okay, so maximum supply of blood will promote more respiration, that is the exchange of gases. Okay, soft होती है टेस्ट and 4 to 8 millimeter thick, leathery and translucent. Translucent is transparent is something that can pass light completely. Now translucent is the substance through which light can pass partially. So this one is translucent in nature, almost transparent in young stage. Almost transparent होती है that complete so that complete light can entire light can pass through it, but in adult stage it becomes opaque क्यों? क्योंकि थिकनिंग की वजह से थिकनिंग हो जाती है तो वो ये ओपेक हो जाती है. It is not transparent anymore. Okay, wrinkled in appearance with a number of folds and depressions all over the surface of body. Now this test, it is made up of a substance which is known as tunicin. वेयर्स ऑफ कम्प्लीटली फ्रॉम द आउटर सरफेस, ये जो इसका आउटर सरफेस होता है यहाँ से ये वेयर ऑफ करती है, और इनर रीजन इसका होता है मेंटल रीजन, जो कि इसका सपोज दिस वन इज मेंटल. ओके, एंड इट हैज़ an outer covering of it has outer covering of epidermis. This one is the epidermis of mantle. Now this epidermis secretes the test gets worn out from the outside, it gets worn out from outside. Okay, and gets secreted by the epidermis. This one is the epidermis of mantle. Okay, so epidermis of mantle which will secrete the test, and it gets wear off completely from the outer surface. Now let's move to the next slide. Test is composed of following components. Now this one is the matrix. Matrix is the gelatinous substance of the test. Matrix is gelatinous substance of the test. So inside the test is present. Suppose this is the gelatinous substance that is present all inside the test. Okay, so this is the matrix, that is gelatinous substance made up of polysaccharide called tunicin. Okay, matrix is sorry not test, it is a gelatinous ground substance made up of a polysaccharide called tunicin, similar to plant cellulose, some proteins and minerals. This tunicin is similar to plant cellulose. Matrix is made up of tunicin, or it is made up of a polysaccharide which is known as tunicin, and it is similar to plant cellulose. So now these are the corpuscles. Mesodermal in origin होते हैं ये corpuscles, that is cells. Certain types of eosinophilus and various small or large cells are present here. 6 to 7 different types. Amoeboid cells होते हैं, eosinophilus cells होते हैं, और the small vacuolated cells हो गए आपके. Receptor cells are also present in this matrix. और nerve cells भी होते हैं which makes test a receptor organ, accessory receptor organ. Okay, so next is interlacing fibrils present होते हैं. Interlacing fibrils का क्या काम होता है? These make a sorry, these fibrils make a mesh-like structure. So this is the mesh-like structure which is formed by these fibrils. They form a mesh-like structure like this, a fine network of fibers. These are the interlacing fibrils that are present in test. Okay, some of them like smooth muscle fibers while others resemble the nerve fibers. Smooth muscle fibers हमारे कैसे होते हैं? These are the smooth muscle fibers which are non-striated. Okay, so some represent this and the other represent the nerve cells. Okay, other represent the nerve fibers. Blood vessels are present from anastomosing system in form an anastomosing system in test. Numerous branches near the surface form oval or pear-shaped terminal knobs known as ampullae. This I have already told you that suppose this is my blood vessel. Okay, suppose this is the blood vessel and the end region forms a knob-like or pear-like structure, and this structure is known as ampullae. Okay, so these are the vascular ampullae that are terminal knobs present here. These are the terminal knob-like structure which are present in the terminal region of blood vessels. Okay, so this is the diagram following the explanation. If you see here, these are the vascular ampullae. Okay, these are certain knob-like structures which are present here. These are vascular ampullae. Okay, these are test vessels. टेस्ट वेसल्स हैं जो कि टेस्ट में प्रेजेंट होती हैं, और इन वेसल्स की टर्मिनल एंडिंग, इफ यू सी हियर, दीज आर द वैस्कुलर एम्पुले, द टर्मिनल एंडिंग ऑफ blood vessels. These are the terminal endings. Okay, and the next one is matrix of test. Matrix of test, this is the gelatinous matrix which is made up of a polysaccharide which is known as tunison. Okay, these are the spicules that are present. We'll study further. Microscleres है और ये spicule sheath है, and these are megascleres which are the large spicules, and these microscleres, they are small in size. Okay, this one is the different types of cells found in test. Okay, so if you see here, I told you large and small eosinophilous cells. This one is the large eosinophilous cell. This one is the large eosinophilous cell. This one is a small one, small eosinophilous cells. This one is amoeboid cell. This is amoeboid cell, and these are the small nerves that are present in test. Okay, यही नर्व्स जो होती हैं ये प्रेजेंट होती हैं और ये टेस्ट को बनाती हैं एक एक्सेसरी रिसेप्टर ऑर्गन. ठीक है, ये हो गई स्फेरिकल वैक्यूलेटेड है, ये vacuolated cells and spherical cells. These are granular cells. Minute granules are present in this, and this one is the nucleus. Okay, squamous epithelial cells are present. Squamous epithelium, like this. Wait. This is the shape of squamous epithelial cells. Epithelium present होती है, उसमें ये squamous cells होते हैं. So this one is squamous epithelial cells. Like this, they are present in test. Okay. और ये nerve cells हो गए, as I have already told you. If you didn't want to take down the notes, then you can pause it and take down the notes. Spicules are present here. Spicules, large number of calcareous spicules, being several equidistant rings, bearing several equidistant rings of minute spines, all pointing in the same direction. Okay, spicules are present here, which are calcareous in nature. It is made up of calcium carbonate, bearing several equidistant rings of minute spines, all pointing in the same direction. Okay, so let's see here. Suppose these are the spicules, like this. Okay, so they bear equidistant rings कैसी होंगी? Let's imagine here, this one is the axis of the spicules. Okay, ये एक axis है और इसके axis के around ये small ring like structures हैं which are equidistant. Okay, equidistant है ये circular rings which are present in spicules. Okay, and these rings, okay, this one is the equidistant rings are present here, and if we zoom a single ring here, this one is the circular ring which bears spines, and spines which face, wait, these are the spines these are the spines which face in the same direction, that is towards the center. All around the ring, these spines are present which face towards the inner side. Okay, so this is the structure of a circular ring which is present in the spicules. Okay, is it all right? See here. ठीक है, ये हो गया था लार्ज spicules bearing several equidistant rings of minute spines, all pointing in the same direction. Now, these spicules are of two types, megascleres and microscleres. As I have shown you in this figure, these are the microscleres. These are the microscleres, and these are megascleres. These are microscleres, and this one is megasclere. Okay. So let's see here. Megascleres are of two types. Pipette shaped होते हैं एक, और एक spindle shaped होता है. Now, pipette. If you have seen a pipette, let me show you here. Okay, so this is the shape of a pipette. This one is pipette. So megascleres are of two types. This one is the pipette shaped. Pipette shaped, which have the middle region curved, slightly curved, and the outer region is pointed. Okay, so these are the pipette shaped, and this one is spindle shaped. Spindle shaped is something like this. Okay, so this one is pipette shaped, and these are the spindle shaped. So megascleres, large sized, occur in all parts of the body except heart, and they are pipette shaped. Okay, straight or curved with pointed ends, each having larger spherical swelling in the middle, reach up to 3.5 millimeter in length. Okay, I have already shown you pipette shape. What is a pipette shaped cell and what does it mean? Okay, so they have each have a larger spherical swelling in the middle. This was the shape. Okay, so they have a spherical swelling in the middle and pointed anterior and posterior, and spindle shaped more abundant होते हैं ये, usually present in bundles equipped with 20 to 60 equally spaced rings of spines, reach up to 1.5 to 2.5 millimeter. And next are microscleres, which are very small in size, 40 to 80 microns long, and confined to test only. These microscleres are only present in test. These are confined in test only. Knob like head होता है and tapering body, beset with 5 to 20 equidistant rings of spines. Rings of spines, what are rings of spines? I have already told you. And let's see this. These are tapering body. They have tapering body. Okay, this one is tapering body and a knob like head. Okay, they have a knob like head with a tapering body. This is known as tapering body with a knob like head. This is the tapering body. If you see here, tapering means जो ऊपर से broad हो और ऐसे नीचे जाके pointed हो रहा हो. This one is known as tapering. So they have tapering body and a knob like head. Okay, that's all. Knob like head and tapering body beset with 5 to 20 equidistant rings of spines. Now, these spicules provide an internal rigid supporting framework like endoskeleton. Keep the mantle firmly fixed to test. Stiffen the walls of blood vessels to prevent their collapse. So इन spicules का क्या काम होता है? फ्रेमवर्क की तरह एक्ट करते हैं, लाइक एंडोस्केलेटन की तरह एक्ट करते हैं. These spicules act as a framework. फ्रेमवर्क क्या हुआ? जैसे कि आर स्केलेटन, ह्यूमन बॉडी में जो स्केलेटन होता है इट एक्ट्स लाइक द फ्रेमवर्क ऑफ द बॉडी। स्केलेटन पे हमारी मसल्स होती हैं और उसी से हमारी बॉडी में शेप आता है। अदरवाइज अगर स्केलेटन ना होता तो वी वुड बी लाइक अमीबा, like this, okay, floating here and there. Okay, so skeleton provides the framework of the body, is the framework of the body. Skeleton provides the shape, a perfect shape to our body. So in the same way these spicules act as the um, supporting framework like endoskeleton. Okay, so keep the mantle firmly fixed to the test, stiffen the walls of blood vessels to prevent their collapse. blood vessels को ये prevent करते हैं collapse होने से. Okay, so let's move to the next slide. These are the spicules. Rings of spines, as I've already told you, these are the rings of spine. Rings of spine present all over the body, if you can see here. And this is the spherical swelling here. Okay, this one is the spherical, this is the spherical swelling present, and these are the tapering body ends. Okay, so this one is types of spicules. ये हो गया एक pipette shaped spicule है, and this one is spindle shaped, which is pointed towards the towards both these ends. They are pointed, and this one is microsclere. Okay, C is the microsclere, which have a knob-like head and a tapering body. So this was the diagram of types of spicules present. Now we move on to mantle. Now mantle lies beneath the test, as we have already studied. It is suspended in the test and attached only to the siphons. These are the siphons present here, and this is attached mantle. Okay, let's suppose this one is the mantle and this is the foot region. So this mantle gets attached only to this branchial and atrial siphons. It gets attached only to this region, and else everywhere it is scattered, मतलब it is dispersed, कहीं भी attached नहीं होती है, सिर्फ branchial और atrial siphon पे attached होगी. Okay, and this is not uniform. It is thick and highly muscular and opaque on the anterodorsal side. Okay, so let's see here. ठीक है, अब ये हो गया इसका anterior or dorsal side. Okay, so this one is the anterior side and this one is the dorsal side. This is ventral side and this is posterior side. So it is highly muscular and opaque on anterodorsal side. Anterodorsal side is this one. This one is the anterodorsal side. So it is highly muscular and opaque in this region. Okay, but thin, transparent and almost without muscles on the posteroventral side. Here is the posteroventral side. So it is thin, transparent, almost transparent, without muscles on the posteroventral side. Posteroventral side is the side where the foot is attached to the body. Okay, so it is almost transparent, and because of this the internal organs are quite visible. Okay, so it encloses a larger water filled cavity, that is atrium. This coelom and atrium will study in the next lecture. Okay, so for the time being it encloses a large water filled cavity, that is atrium. For the time being let's only study the term here, and in detail we'll study in the next lecture. Sorry. Mantle. Now this mantle consists of other three layers. This is outer epidermis, mesenchyme, and the inner, it is inner epidermis. Okay, so outer epidermis, mesenchyme and inner epidermis. मैंटल लाइज बीनीथ द टेस्ट, ये सब हो गया है। चलो एपिडर्मिस हो गया, आउटर एपिडर्मिस। आउटर एपिडर्मिस इट इज मेड ऑफ सिंगल लेयर ऑफ फ्लैट हेक्सागोनल cells. At branchial and atrial apertures, it is interned to reach up to the base of siphons and forming stomodium and proctodium. Okay, so here it is. Okay, so made of a single layer of flat hexagonal cells. Hexagonal cells, let me show you here. Okay, these are hexagonal cells. This one is the hexagonal cells which consist of 6 sides made up of a single layer. ऐसी एक single layer होती है hexagonal cells की, like this. Like this. Okay, here they are surrounding the body. Okay, ये कैसी फ्लैट सी लेयर होती है हेक्सागोनल सेल्स की, ऐसी एक लेयर होगी। फ्लैट हेक्सागोनल सेल्स एट ब्रैंकियल एंड एट्रियल एपर्चर्स इट इज इंटर्न्ड, मतलब इंटर्न्ड होती है, अंदर की तरफ टर्न्ड होती है, टू रीच अप टू द बेस ऑफ द साइफन्स। ओके, एंड फॉर्मिंग स्टोमोडियम एंड प्रोक्टोडियम। अब स्टोमोडियम और प्रोक्टोडियम क्या है? स्टोमोडियम एक नैरो ट्यूबुलर सी कैविटी होती है ब्रैंकियल साइफन में। ओके, ब्रैंकियल साइफन की एक नैरो ट्यूबुलर कैविटी होती है दैट लीड्स फ्रॉम माउथ टू एनस। माउथ, ब्रैंकियल साइफन को ही ब्रैंकियल साइफन मतलब माउथ मार्क करता है ब्रैंकियल साइफन को, ठीक है। माउथ प्रेजेंट होता है ब्रैंकियल साइफन में एंटीरियर एंड पे बॉडी के, ठीक है। तो वहाँ से एक नैरो ट्यूबुलर सी कैविटी निकलती है जो कि फेरिंग्स को लीड करती है। एंड व्हाट इज प्रोक्टोडियम? प्रोक्टोडियम एट्रियल साइफन की एक कैविटी होती है जो कि लीड करती है फ्रॉम द एंड, फ्रॉम अ रीजन वेयर रेक्टम ओपन्स। ओके, अब जहाँ से ये रेक्टम ओपन करेगा वहाँ से ये एक्सटीरियर को लेके जाती है। दिस वन इज प्रोक्टोडियम। Okay, so this was proctodium. Now let's move to mesenchyme. Mesenchyme lies below the outer epidermis derived from mesoderm. mesenchyme एक लेयर होती है जो कि mesoderm से बनी हुई होती है and it lies below the outer epidermis. It consists of connective tissue fibers traversed by nerve fibers, muscle fibers, and extensive blood sinuses. Now, as we have already studied, basic structure एक लेयर का यही होता है। आउटर एपिडर्मिस होगी, मेसेनकाइम होती है और इनर एपिडर्मिस होती है। सो मेसेनकाइम इज दैट मिडल लेयर विच इज़ हाईली सप्लाइड विद ब्लड वेसल्स, ठीक है। ब्लड वेसल्स से सप्लाइड होती है। ऑर्गन्स की लाइनिंग, सॉरी नॉट ऑर्गन्स, एक मिड लेयर होती है जो मसल्स, वी कैन से डैट मस्कुलर होती है, मसल फाइबर्स होंगे उसमें, ब्लड वेसल्स होंगी। सो इट्स द सेम केस हियर, कंसिस्ट ऑफ कनेक्टिव टिश्यू फाइबर्स ट्रेवल्स बाय नर्व फाइबर्स, muscle fibers and extensive blood sinuses. Muscle fibers unscripted, sorry not unscripted, it is written here wrong, unstriated होगा ये, let me mark it, this one is unstriated. Unstriated muscle fibers that are arranged in 3 sets. Annular muscles होती हैं, surround each siphon in circular rings. Annular muscles, as the name suggests itself, annular something which is circular in region, circular in structure. Annular muscles होती हैं that surround each siphon in several circular rings. तो ये एक circular rings की तरह surround करती हैं, ठीक है, circular rings. ये प्रॉपर नहीं है, मतलब रिंग भी सर्कुलर होती है, सो इट्स नॉट करेक्ट टू से दिस. Annular muscles surround each siphon in the form of a ring. Okay, cause the contraction of siphon. Rings होती हैं तो contraction easily cause होता है, इसलिए annular muscles are present at the base of the siphon. Okay, so annular muscles followed by longitudinal muscles. Longitudinal muscles start from branchial and atrial apertures and they radiate beneath annular muscles. Okay, एन्युलर मसल्स के नीचे रेडिएट करते हैं टू द मिडल ऑफ द बॉडी, अप टू द मिडल ऑफ द बॉडी ऑन ईच साइड, ब्रिंगिंग अबाउट द कॉन्ट्रैक्शन ऑफ बॉडी. लॉन्गिट्यूडनल मसल्स हो गए. सबसे पहले एन्युलर मसल्स आते हैं जो कि कॉन्ट्रैक्शन कॉज करेंगी, साइफन्स में प्रेजेंट होती हैं. फिर आई लॉन्गिट्यूडनल मसल्स जो कि दोनों साइफन से एक्सटेंड करती हैं अप टू द मिडल ऑफ द बॉडी and they are present beneath the annular muscles. Okay, and after that a few branchioatrial muscles, atrial, sorry, not atrial, a branchio a few branchioatrial muscles extended deeper between the two siphons. Connective tissue cells present in the middle layer are mostly amoeboid as vacuolated cells. Okay, so middle layer में ये जो कनेक्टिव टिश्यू के सेल्स होते हैं दे आर मोस्टली अमीबॉइड एज वैक्यूलेटेड सेल्स. तो ये हो गया ब्रैंकियोएट्रियल मसल्स होती हैं लॉन्गिट्यूडनल मसल्स के बाद. सो पहली थी एन्युलर मसल, फिर आई लॉन्गिट्यूडनल मसल्स और फिर आई ब्रैंकियोएट्रियल मसल्स विच एक्सटेंड डीपर बिटवीन द टू साइफन्स. अब लास्ट है इनर एपिडर्मिस. इनर एपिडर्मिस इज द इनर ectodermal single layer of flat polygonal cells lining the atrial cavity. ओके, इनर ectodermal single layer होती है, that is of flat polygonal cells. Polygonal cells is the lining that forms the atrial cavity. So flat polygonal cells होते हैं which line the atrial cavity. और ये किस में होते हैं? इनर एपिडर्मिस में, that is inner ectodermal single layer होती है वो. Okay, so with this we come to the end of this lecture. Last but not the least, thank you so much for attending this lecture, and if you are satisfied with the content provided, then please like, share and subscribe my channel. Thank you so much for attending the lecture.